the lecture that will take place now precisely wants to contribute to this shared vision around key issues in global health, and therefore we have the privilege today to have Professor Dyann Wirth, who is Richard Pearson's professor and chair of the Department of Immunology and Infectious Diseases at the Harvard School of Public Health, and also senior associate member and co-chair at the Broad Institute in Boston. Let me just very briefly introduce her, although she's a long-standing friend and scientific collaborator with different research teams in Kesehatan. Let me just make a very brief introduction. As I think will be clear, and some of you know already, a major leader in the area of malaria research. So this is a little bit some of the core of our collaboration, and mostly in issues regarding how the parasite has evolved, especially in areas of population biology, drug resistance, and antigenicity. And some of the strengths that are really spectacular and good to share with her here today is the strength and the capacity to set up a very broad multidisciplinary team that includes or that brings together scientists in the areas of molecular biology or genetics, genomics, population genetics, chemistry, cell biology, epidemiology, computation biology, biostats, and also clinicians in areas in infectious diseases. So that means a broad range of disciplines that work together to generate a very intense and fruitful research program. And in this area, since some of her activities have precisely to do with the use of state-of-the-art technologies and novel approaches to, from the point of view of genetics and genomics, to understand a little bit how this parasite works and how this has to do also with its resistance to whatever we try to somehow to control him or to get rid of the parasite. So anyway, it's with great pleasure that we welcome you here. We are aware that the topic we suggest is a very broad one, both from global to genetics, but this adds a special interest to the effort that this means, and thank you for being here. And we agree that this will be rather informal, so there will be the capacity to have questions and interactions. Thank you very much, Dyann.
Thank you. Someone's going to need to organize my slides. Yes. All right. Well, it's truly a pleasure to be here. Can you hear me if I wander around? Okay. It's really a pleasure to be here, and thank you very much for the invitation. I am here to teach in the Science of Eradication course. So we have leadership individuals from around the world, from ministries of health, from NGOs, from sort of industry, from funding agencies, all focusing on the sort of science of how we're going to get to eliminate malaria. And so it's been fascinating all week, and that group has been very lively with discussions, and most people who've been lecturing there haven't been able to finish their slides. So that's the goal here, is we should have a lively conversation. So what I'm going to talk about today is work that my group's been involved in over the last now 6 or 7 years to really take the approaches that were pioneered by the Human Genome Project and apply those approaches specifically to the parasite. I'm going to talk mostly about Plasmodium falciparum today. I know Ivo loves Plasmodium vivax, so I've put a Plasmodium vivax slide in. We are beginning now to apply these methodologies to Plasmodium vivax, and I think you'll be able to see the kind of opportunities and the questions that we're able to ask in with this kind of new knowledge. So I'm going to go through, I'm going to focus on malaria, but I think you can see as I talk, and I'll try to point it out, that there are opportunities beyond malaria to other infectious agents. And I think one of the really exciting things about the field right now is that we're able to look at infections in their natural situation, okay? So we're actually able to study at a very fundamental level, at a very fundamental genomic level, the interactions between host and parasite in this case, or mosquito and parasite, but that can apply to almost any infectious disease. And I predict not only is there going to be an explosion of knowledge, but there's also going to be an explosion of translational insights that we didn't have before. So I'm going to try to illustrate that with malaria. I'm going to give somewhat of an overview of what we're doing, and we probably won't get to the last set of slides, but I'm happy to talk to individuals about different aspects of the project as we go on. Okay, I never get to the to the acknowledgement slide, so I've started to start with the acknowledgement slide. So this is really, most of what I'm going to talk about today is a collaboration between the Broad Institute and the Harvard School of Public Health. Really the key leaders of this, Eric Lander invited me. Eric is the director of the Broad Institute, and many will know his name from the Human Genome Project. I was trying to get the little, well, it's okay. And he invited me when he formed the Broad Institute to come and really start looking at infectious diseases from this genomic way. So Eric is on the right in the top set of people. Sarah Volkman and Pardeep Subedi really have been the two local leaders in this project, and Dyann, who's fourth from the left in that first row of people, really has also taken on, in fact, in some ways, has taken on the major leadership role from the genomic side. And yeah. I want to try this. All right, that's okay. Somehow this worked earlier when I tried it. Oops. That's okay. I don't really need it. So this takes a big team. There's also, I'm going to talk about work in our collaboration with the group at Cheikh Anta Diop University in Senegal. Dauda and Dyann is now the senior person in charge of that group. And we've transferred a lot of technology and knowledge to that group. And Dan Hartle, in the bottom left-hand corner, is the population geneticist we've been involved in. So there are lots, I'll try to acknowledge people as I go along, but this takes a big group, and it's been a very interactive group. Okay, this is kind of an overview of how we approach applying genomics, in this case to malaria. It's there, okay. Oh, perfect. So we start with now patient sample collection in the field, okay? So we can take a sample from a patient. So that's what I mean by really understanding this really at the natural level. We can directly sequence, we now just directly sequence from the patient sample, the parasite genome or the human genome. We can genotype, I'll talk a little bit about that. We can determine the phenotype in vitro, or we can have a phenotype from a clinical presentation. We can scan the genome looking for signals of selection and generate those. We can do genome-wide association studies. We can have parasites with a phenotype. An example I'm going to give you is drug resistance and drug sensitive, and ask what genes, what parts of the genome are associated with this phenotype. So again, directly adapting the technologies developed from the Human Genome Project. And then we're developing assays, and I'll show you a couple of examples of these, that you can take, maybe not right to the field, but certainly to labs set up near field stations, and I'll give you a couple of examples of that, with the application really for drugs, vaccines, and understanding population biology. So this is a sort of pipeline approach. It's a way of thinking about it, and it's a way to generate enormous data. The other thing that's happened in the 10 years since the first Plasmodium falciparum genome was sequenced, or the first human genome was sequenced, is that the cost of sequencing has fallen dramatically. And so you're down to a human genome costing $1,000. A parasite genome now costs about $100 to sequence. The first one costs $30 million. So this allows us to really generate an enormous amount of information. But of course, that information alone isn't valuable unless you can connect it to the biology and the disease. And I think that's the challenge. And what I would say is we're right at the beginning of that. So we now have, we can generate as much information as we want, but now I think the real question is, what questions do you ask and what do you get out of that data once you have it? And I'll show you some examples where I think we're beginning to get some insight into parasite biology. So much of this is published, and so I won't spend a lot of time on it, but as with human populations, you can do straightforward population biology on the parasite. This is just showing one example. This is a principal component analysis of some sequenced parasites. And so we can see that, for example, the parasites from the Americas and from Asia are quite different than the parasites from Africa. So in fact, if I sequence a new parasite, and I haven't been told ahead of time where that parasite comes from, I can actually just plot it into this figure, and I'll be able to tell you. And also you can see that you can almost get a sense, and this is actually a geographic sense, as you move further away from Africa, the parasites are more different from the African parasites than the ones that are closer. And this is almost a geographic, it's not quite a linear line, but it probably gives you a sense of how, in this case this is Plasmodium falciparum, how the parasites originated in Africa and then migrated as individuals migrated to the Asian subcontinent and the South American continent. And so this gives us a real sense, and it allows us practically to tell where a parasite comes from. So for example, as we move to the elimination of malaria, one of the real questions, as you get down to very low or no transmission in the country, then the question is, if a malaria case appears, is it from the parasite that was originally there, or did someone come in with a parasite from elsewhere in the world? This methodology will allow you to directly assess that. So it has practical applications, but it also tells us about the biology. The other interesting thing, and we'll come back to this a lot, I just want to point this out here because it's the only place I have an LD plot. If we plot linkage disequilibrium, let's just set this up. I actually should put a human on here. So if you measure the average linkage between human genes, it's about 2 million base pairs, I would say, 200,000 to 2 million base pairs. Well, in the parasite, it's about 500 base pairs in Africa. So what that means is that the parasite is recombining very often and with different parasites. And we'll come back to that. Africa, and this is directly related to the transmission and the multiplicity of infection. And we'll come back to this a little bit later. But in Southeast Asia and South America, LD is significantly longer, and this is consistent with there being fewer parasite genomes there, fewer chances to exchange. And this information, again, very fundamental science, actually is very practical in actually telling us what's going on as the parasite is being transmitted. Okay. All right, so just to give you a few statistics, the parasite genome is about 24 million base pairs. So this is a really serious organism. It's about the size of yeast, okay? It's about 100-fold smaller than the human genome. And it has 5,000 genes, it has 14 chromosomes, it's haploid, and we have sort of all of the tools we need to examine it. We have the genetics of the parasite, and we're able to examine expression, RNA expression or protein expression. So we can really interrogate this organism at very fundamental levels, and I'll show you some data from all of this. So the first thing I'm going to talk about, in part because I think for those of you who are familiar with the vaccine studies that are ongoing in malaria, you know that the vaccine doesn't work 100%. And so one of the things we've been interested in is using genetics to try to understand vaccine efficacy, or to try to understand what's going on with vaccination. And we didn't start out here. I would say this is like an idea that came after we saw the data. So when we sequenced the first 20 or so parasites, so this was in the early days when it was still $100,000 to sequence a parasite, so we only did a few, we made an interesting observation. Now, had I been asked to make a prediction about the parts of the genome or the genes within the genome that have the highest diversity before I saw this data, I would have argued that it would be in the drug response genes, right? Drug resistance is a big problem. We'll come back to that a little later. But in fact, it's almost all the diversity is in genes that are expressed at the surface of either the infected red cell or the parasite. Now, why is that? Anyone want to guess? Do you know? I see a smile. But he knows the answer. Why is it? So this is the entire genome. So this is all 24 million base pairs sequenced across parasites from around the world. And we find the highest number of variants, single nucleotide polymorphisms, in genes that are expressed on the surface of the parasite or on the surface of the infected red cell. What? Immuno, yeah, right. It's immune selection. And for those of you who know me, a subset of you in the room know me, I used to always say, immunity is not an issue in malaria. It's not going to be important. Well, it turns out it is the single most important driving force represented by the footprint in the malaria genome. 90% of the single nucleotide polymorphisms in the parasite genome are likely to be driven by selection by the host immune system. So it's an incredible selective force. Drug resistance, differences in metabolism are a minor set, not an unimportant, but a minor set in terms of numbers. The other important observation is that this is just another way. This is these are the Go functions. These are the functionalities. I don't expect you to read this. This is just the Go functions arranged high diversity, the number of genes with high diversity in this functional category is represented in red, and with no diversity is represented in black. And I will tell you that all of these very predominant high diversity gene classes, in fact, are represented by these genes that are on the surface. Genes for intermediary metabolism, DNA replication, are all down in this black section. They're completely monomorphic worldwide. Now that's, as we get hundreds of thousands of sequences, perhaps that's not going to hold, but these are very conserved genes. But the interesting thing is that, in fact, there's this very long tail of parasites, of parasite genes that have extremely high nucleotide diversity. Now, if we get down to practical things, these are actually the vaccine candidates. The circumsporozoic vaccine is the vaccine that's now in phase 3 trials. These are 3 surface antigens on the merozoite. These genes show a higher nucleotide diversity than the most diverse human gene, which is the human HLA antigen family. So what this tells you is this is very unusual. Now, that means that, at least in my mind, we need a completely new paradigm for thinking about the parasite and its interaction with the human host. What do you think this means for a vaccine for this organism? Did I hear something? What? It's not going to be easy. Exactly. In fact, what other organism is kind of like this? And what is a vaccine? What other infectious organism shows what? I think I heard it.
Influenza.
Influenza, exactly. Right. So influenza, and believe me, if I plotted the influenza nucleotide diversity on this, it wouldn't look anything like this. The influenza diversity is kind of down here below HLA. So these are much more diverse. And what it tells us is the parasite, in its existence with the human immune system, the mechanism this organism seems to have evolved is a high number of diverse genes that are expressed and present to the immune system, essentially a novel antigen each time there's an infection. I also think this is the explanation for there being no sterilizing immunity by natural infection in this infection. Okay. All right, so we can, of course, look at this in more detail. This is just plotting of the data in another way. This is a slightly larger data set. But we can plot, in this case, this is diversity in Senegal. So we had 45 strands from Senegal versus Senegal versus Thailand. So this is divergence. So within a population and between populations. We expect within a population to see diversity primarily in things that are responding to the immune system. And if we look at what these are, they're all antigenic variations, cell adhesion, cell surface. So again, exactly the same concept. And these are just examples. Those of you who work in malaria will recognize the genes. Many of these have been touted as vaccine candidates. I would posit that any of these will not work as a vaccine candidate because pre-existing in the population, in the extant population of parasites, are already parasites that will not respond if you vaccinate with a single allele. So very similar to influenza. You either have to think about a polyclonal, sort of a multiple allele vaccine, or some other strategy, which I'll talk a little bit about that later, if there's time. Okay, so we're looking at genes under immune selection. The other thing we can look at, and one of the other fascinating things about this organism, is that in fact the genes are not only diverse in their sequence, but they're also diverse in their expression. And so we did some expression profiling. I'm just going to show you an example. So here are some genes that we identified as potentially being interesting based on their expression in patients. But here is their expression in 3D7. This is the parasite we grow in the lab and upon which almost all existing molecular biology expression analysis has been done. But here are examples from patients. These are 20-some patients, 25 patients from our cohort in Senegal. And you can see these same antigens. In some cases, this particular antigen is expressed in almost all of the patients, and it's not expressed at all except at the merozoite stage in the in vitro cultured parasite. So this is the first kind of critical lesson here, that is understanding parasite, not just biology, but even something as fundamental as expression. It's different in patients than it is in long-term in vitro cultured parasites. And I think this is extremely critical to understand if you're going to think about vaccine development. None of these molecules would have been identified if we were working only with the 3D7 parasite. We also can, just to show that we can really find these are direct patient samples, and then we cultured patient parasites from patients in vitro, and we're able to show that a number of these genes continue to be expressed in early culture, but as we move to extended culture, if you just look at this gene, 950W in this case, just as an example, it's expressed for the most part in the parasite strains in early culture, but then as you get to late culture, it turns off, similar to what we've seen in 3D7. Another example, 14755, expressed in most of the early culture parasites and turned off. So I think that, again, this is a very important concept which genomics led us to, and I think it means that studying infections in their natural state is going to be critically important. Okay, so one of the things that we're particularly interested in, and for those of you who know me, I'm not an immunologist, but this data sort of drove me to start thinking about vaccines and vaccine development. So, of course, the idea is to use this information as potential target identification. And so our strategy is actually the opposite. We are concentrating on the monomorphic genes, those genes that show little or no variation, including genes in these classes expressed on the surface, with the idea that these are genes that perhaps are not under natural selection. Perhaps these are genes either where variation will cause a fitness loss, or that these have not really been subject to human immune pressure for whatever reason. They're masked or whatever. So this is a strategy. We have a collaboration with Sanofi Pasteur in Boston. And I will say that there are actually a number of very interesting monomorphic genes which we're selecting and beginning to analyze. Obviously, we'll have to validate them. We'll have to know if there's human response. We'll have to test their function. But I think it's a completely different idea. It's now sort of turning it on its head and saying, okay, instead of choosing polymorphic genes, choose monomorphic genes as your vaccine target. And I think that's a conceptual piece which many different, or it could apply to many different organisms. Yep.
Sorry, but I don't understand this approach, because if these antigens are not exposed to the immune system, so what will be the value of a vaccine based on?
Well, you know, exposed, they might not be antigenic, right? We don't know that they're not exposed. What we know is that there's no variation in the natural population, right? If they're essential genes that have an essential function, it may be that the way the parasite has evolved, one theory, I mean, I agree, you know, it's a hypothesis, but one theory is that the genes that show massive variation, in fact, are almost a smokescreen, a way for the parasite to protect key functional epitopes, whether those are whole proteins or monomorphic regions within otherwise variable proteins. And you could apply it both on a whole gene basis or on a subregion basis. Because otherwise, I mean, I think if something like this doesn't work, my feeling is that the chance of making a vaccine is quite doomed, because I think to make a polyvalent vaccine that would cover enough of the world population of parasite diversity is almost impossible. And we've done calculations that you might need 30 different epitopes in a vaccine to get coverage of enough of the antigenic diversity. So the alternate strategy is to say, perhaps some of these monomorphic genes or monomorphic regions, in fact, are in some way being protected by those regions that are varied. But again, you know, hypothesis. So we will pick things that have function, either in invasion or in metabolism or transport. And we actually think that might be a very interesting place to go. But it's a great question, and it's definitely controversial. Okay. All right. So I'm going to turn now to talking about. So this is the first life cycle science. You know, I rarely give these talks. I always have to have a life cycle science somewhere. But this one, and you've probably seen this before. This is actually a slide that originally was designed by Bob Sinden. So I'm now going to talk a little bit about population biology and how we can use population biology to inform sort of about the real world presence of the parasite. Okay, so there are a couple of things I want you to think about. First, so this is now the life cycle kind of in a linear way, and it's entirely focused on the parasite. So this is humans, mosquitoes, and then back to humans. So if we start with the liver schizont, okay, this liver schizont for Plasmodium falciparum releases in the end about 10,000 parasites. Those parasites invade the red cells, and the asexual parasites asexual parasites reach about 10 to the 11th per person. This number varies a little bit, but maybe in some cases gets up to 10 to the 12th per person. But it's a very large number of organisms. Then there's a replicative cycle, but a subset of the parasites differentiate into the version which can be transmitted. These are actually the biologically critical part of the cycle. Without gametocytes, male and female, remember I told you the organism is haploid, but every genome, every parasite genome can form either a male or a female gamete. You can form both a male and a female gamete, and that's important for the next discussion. When these just float around in the blood until the mosquito takes a blood meal, when you get fertilization, so sexual exchange at every transmission, formation of the macrogamete, migration across the gut wall to the oocyst, release the oocyst, these are the sporozoites from the salivary gland, these invade the human and the parasites go back to the liver. So that completes the cycle. So I want you to notice a couple of things if you think about population biology. There is a bottleneck twice in every life cycle. One bottleneck is a little tougher. This is that only 2 to 5 genomes actually successfully get transmitted in nature. Maybe a little bigger in some cases and a little smaller in others, but it's a very small number compared to 10 to the 11th. Okay, and then coming out of the mosquito, you're probably in the sub 100. And again, there's controversy and it may be variable, but it's certainly much smaller than 10 to the 11th and in the same, maybe slightly larger order of magnitude. So this is a very important concept. Yeah.
I'm wondering in this, because we've been involved Sinton talk about this as well. There is one slight logical error in here, in a sense that, not that it goes up to 10 to 11. It's not that one human infects one mosquito and then one mosquito bites one human. One human infects potentially a number of mosquitoes.
Right, but it's still a small number.
It's a smaller number, but I think it is.
But it's still, I mean, in order to make, even if 100 mosquitoes bite, it's still a very small number.
Much smaller than 10 to 11.
Oh, much, much smaller. So, I mean, even if 100 mosquitoes, and it's probably rare that 100 mosquitoes bite and get infected from an individual, but nonetheless, it's still a huge bottleneck. It's several orders of magnitude of a bottleneck. Okay, and similarly here, most people are bitten by one mosquito at a time, one infected mosquito at a time. So again, from a population biology standpoint, these are bottlenecks, and these don't occur in most organisms. This kind of population bottleneck. So it's very important when we think about population biology, and it's going to be very important when we think about the genetics. Okay, so we're going to skip over that because of time. There's another bottleneck, okay, and that occurs in a much more evolutionary time frame. So Plasmodium reithonaui, okay, is the parasite that is most closely related. It's a primate parasite, and one can estimate the divergence of reithonaui from falciparum. What's important for this is that at some point, and we're seeing this today with Plasmodium knowlesi in Southeast Asia, and we're seeing it with influenza, avian influenza virus, at some point the parasite entered humans, right? It probably entered humans many times, like hundreds of thousands, millions, maybe billions of times. But at some point, there was a successful establishment of infection and transmission, human, mosquito, human, okay? With Plasmodium knowlesi at the moment, it's coming from a primate going into humans, but the transition of human to human mosquito, not human direct to human, but human infected, mosquito bites human, and that parasite is transmitted to another human, has yet to occur in Plasmodium knowlesi. But the hypothesis is that it occurred in falciparum. And, we've been back and forth about when this occurs. Our current data, looking at now several hundred sequenced strains from Senegal, is that there was about a 60-fold population expansion about 40,000 years ago, 20 to 40,000 years ago. So there was this successful expansion into humans. And then humans migrated, infected humans and their parasites migrated to the continents. And all of this time, there's been selection on the parasite by the human immune system. Very recently, relatively speaking, there's been selection in drugs. And now in 2007, I'm going to show you that in fact introducing insecticide-treated nets, careful diagnostics or accurate diagnostics, and effective drugs is probably producing yet another bottleneck. So what's happening, so you think about it, evolutionary time, these parasites are the survivors, and they're about to undergo another very strong effect. So you have two bottlenecks, one every cycle that's biological, and this more evolutionary scale, one of which we think we're seeing evidence for this actually being a strong selective pressure right now. Okay, so now we're going to come back to this idea of recombination and linkage disequilibrium. And what I'm going to talk about, okay, is a tool that we've developed which allows us to look at the genome in a high throughput, relatively low expense way, but reflect the overall population compared to the choice of sequencing. This is what we call the molecular barcode. It detects single nucleotide polymorphisms at key regions along the parasite genome. Each and every genetically distinct parasite has its own barcode. So there are 24 single nucleotide polymorphisms in this particular tool, and this is the one I'm going to tell you about today. But we do have a new tool now that has a few more, and there's a reason for that. But each parasite has its own barcode because at each position there are two choices. It can be either a C or a T. So these parasites differ at this position. These parasites are identical at this position, and so on. Statistically, this is the chance of having this same barcode appear in two independent parasites is 1 in 2 to the 24th. So it should never happen. Okay, so now I need to tell you a little bit about the Plasmodium life cycle, in this context. Okay, so here we have a situation where, as Eva was saying, people can either be infected with a single parasite or with multiple parasites. Okay, it depends on the frequency of transmission. It depends on what the parasite population is, but this occurs. And in fact, this number can be larger. It could be 1 or greater than 1. Okay, in order for there to be diversity, in fact, this condition has to exist. And let me show you why. If you think about the mosquito midgut, this is for those of you who work in simpler model systems, you can think about this as a yeast cross. So if a mosquito takes a blood meal and there are male and female gametocytes, there's going to be male and female gametocytes from each of the genomes if you have a mixed infection. Okay, so you get outcrossing and you generate sort of a new barcode because you've reassorted the chromosomes and you've had recombination. So each progeny, so you get pairwise, you get 1 male with 1 female, and that progeny will have a unique barcode. So each oocyst will actually have 4 types in it, just like a yeast tetrad. So that will generate 4 different genomes for the next round of infection. So if you had, if you have 2 parasites, you get 8. If you have 3 parasites, you get 16. I think you get 16. Okay, so you can see this is like really goes very quickly, and you generate diversity. Okay. On the other hand, if you're in a situation where transmission is actually quite low, and the chances that someone's infected only with a single parasite, okay, the same thing happens, the same biology happens, but every parasite that comes out is identical. Okay, so this is a tool that actually allows you to measure outcrossing, and so we've done that. Let's see what the next slide is. So here is an example. These are from Senegal, and these are just parasites. These are, let's see, the positions across here, and the parasites down here. And bolded, you can see, so for the most part, these parasites from patients are in fact unique, except you notice something. Here's a parasite that has exactly the same barcode as this parasite down here. And this is Africa, so this is from Chess, Senegal. This is an area of reasonable transmission. So we were quite surprised at this, and believe me, we spent a lot of time demonstrating this wasn't just, a family that was infected by one mosquito that happened to be particularly good at transferring blood meal. These are all different individuals spread over time in the population. That observation was actually made in 2008, and we had 22 parasites that were identical to one another in different people in the population. But we also had other parasites that had either two partners or one additional person infected. And if we go, and so then we went back and looked at the data before 2008, and in fact this phenomenon was there, we just never noticed it because we didn't expect it. And so as we go through time now, we have data down, well, we actually have the 2012 data, but it's not on the slide. You can see actually that the proportion of repeated barcodes is increasing, consistent with there being a bottleneck. Another way to look at this is looking at the proportion of single or unique infections in the population, and you can see going from 2006 down to 2011, we are slowly but surely, and in almost a linear way, going to these clonal, small number of parasite genomes in an African setting. Supporting this, if we now look from year to year, this is now, and I apologize, this is the one slide that looks like it didn't translate, and I didn't notice it. So this is 2006, 7, 8, 9, 10, 11. Okay, these are genomes, and the size of the circle represents the number of genomes. So in 2006, there was one genome that appeared in 2007 and actually increased. In 2007, there was a single genome that was carried over. As you start to look in 2008, this is the large 22 patients that had the same genome. It appeared the next year a smaller number, but here you see there are actually 3 parasites that reappear. Here in 2009, there are 1, 2, 3, 4. In 2010, you're starting to see the picture. So what's happening is the diversity of this population is decreasing. Okay, well, what could that mean? Well, Senegal is in the situation of having introduced RDTs, rapid diagnostic tests, ACTs, insecticide-treated nets, and increased IRS during this period, starting sort of the 2006, 2007, and continuing. Is that the cause? I have no idea, but it's very interesting that we're starting to see an effect on the parasite population coincident with introducing much more pressure on the parasite. Okay? We're reducing transmission by bed nets and by insecticide spraying, and we're killing the parasites with ACTs. So I think this is very interesting. We see a similar phenomenon, although we do not have longitudinal data. And so with a real grain of salt, this is Panama. This is a collaboration with Nicanor Obermann at the Gorgas Institute. And here is just a slightly different way of presenting the data. He has typed 2 parasite, he's typed all of the parasites collected by the National Malaria Control Program during the year. Malaria, so this is Plasmodium falciparum. Plasmodium falciparum is almost eliminated in Panama. When he did the barcoding, he found 2 different identical clones, and then he found some outliers. all of these are imported cases, and all of these are indigenous Panamanian cases in 2 foci. So again, a very strong bottle, sort of a clear indication that the population biology of this area, these parasites, is really quite different than in a highly endemic area. So we actually think this now provides a tool for determining transmission. That actually this tool, this appearance of clonal parasites or a change in complexity of infection, another sort of application of this kind of tool and other approaches, will actually tell us what it's related to the transmission and specifically related to the amount of outcrossing. So we think this is actually a new way of measuring transmission where you don't actually need to measure the infected mosquitoes or biting rates or anything. You just look at the progeny of the outcross, which are the individual parasites in the person. And so again, this is going to be a little controversial, and it'll be something where it won't be completely accepted. But I say this because I actually think this is really fundamentally, it's fundamentally just detecting a genetic cross. That's all transmission is. It's just a genetic cross. It's either self or outcrossed. There are no other possibilities, and there are only 2 genomes involved in any one of those events. So if you think about it, it's a very simple, fundamental concept, and then one needs just to integrate this with other kinds of measurements. So I think we see something very interesting here, and we hope that this is going to turn into a tool. So the idea is, in very high transmission areas, you have people with lots of different parasites. As you move to moderate transmission, you start getting people with clonal parasites, some mixed infections, and then at low transmission, you start getting these repeat barcodes where a single parasite or a small group of parasites seems to survive. This may also give us a little hint that there's going to be parasites that survive best under those conditions selected. Okay, so again, another sort of thing worthy of investigation. Okay, all right, we can obviously apply this approach to all sorts of things. This is just looking now a little bit more practically. Can we follow, for example, the appearance of drug resistance in population? And I show you this data. This is part of the high-resolution MELT technology, which we've developed and is soon going to be introduced in Mahisa, to be able to detect individual mutations. So this is down to detecting a single point mutation, but in a very high throughput and relatively inexpensive way. And this is just from some of our work, again, the same population from Senegal, but this time we're looking at specific alleles that, in this case, we know are associated with drug resistance, as they change in the population over time. And what struck us is 2 things. This is the PFCRT, this is the gene associated with chloroquine resistance. Chloroquine is no longer used in Senegal, yet we seem to be seeing a change in one of the alleles in this gene. It's increasing its prevalence in the population from 2008 to 2010, whereas other genes in the PFCRT allele are pretty much flat. But if you look at PFMDR1, this is the major drug transporter associated with methicillin resistance. This allele seems to be going almost to fixation over this period, and this allele, these 2 alleles are going almost to extinction. Okay, so this is like selection in real time. This is looking at all the population of parasites and asking what's the allele frequency. All right, so what could this be? What's one explanation? What happened in 2007, 2008 in Senegal? It's when ACTs were introduced. We think this may be indicating a response, a population response to the introduction of artemisinids. We know from in vitro work in Alan Cowman's lab that this gene confers a really low level of resistance to the artemisinin compound. We also know from our own work that lumefantrine, which is the partner drug now in Senegal, is actually resistant to lumefantrine, or sensitivity to lumefantrine is mediated by this gene. So we actually think we're seeing an early warning of a change in the population in response to this pressure. Again, this is hypothesis. We are now transfecting, making transgenic parasites to look at this in a much more detailed way. The other sort of approach that we're taking to look in more real world, this is a collaboration actually here with the group at CRESEB and now with the entire RTSS vaccine group. The idea is to actually look at the target of a vaccine to see if after vaccination there's a change in the parasite population, the alleles that are targeted by this vaccine. Remember at the very beginning I told you that in fact I thought almost all of the diversity in the parasite, or 90-some%, 80 to 90% of the diversity, is driven by immune selection. The antigens, the current genes that are targeted, the alleles or the proteins, or parts of proteins that are being used in vaccines, fall into this category. The CS protein is one of those. This is the protein in the RTSS vaccine. It has a polymorphic region. And so one question is, if you vaccinate, can you in fact, do you start to select the alleles that are not protected, okay? So I don't have an answer for you, but this is the technology. So this again requires sort of new high throughput sequencing, next generation sequencing. So what we do is amplify this region, and then in a sort of massively parallel way, we sequence from hundreds of patient samples all of the CS, both in vaccinated or unvaccinated. And we can actually deconvolute and tell which are the sequences, the polymorphisms, and what the numbers of those alleles are in every patient. So again, this is exactly like looking for a drug resistance marker. This is looking for a change in the population after vaccination. And obviously, that trial is still ongoing. We do not have, although we do now have samples, we don't have, the code is not broken, so we won't know what the answer is. But I can say that I actually think this is going to be, again, extremely powerful, not just for Plasmodium falciparum, and in fact, perhaps even more powerful for HIV, particularly for HIV, and perhaps for influenza. Okay, so this is the description. So basically, it's to apply genomic approaches to evaluate the outcome of the RTSS. And the concept is the parasite that appears the same or different after vaccination? And in fact, do vaccines act like drugs and select a few escape variants? So that's the question. I don't have the answer, but I thought you would enjoy hearing the question. Okay, I'm going to end for Evo on Plasmodium vivax. So Plasmodium vivax, the forgotten parasite. We now have started, it's been a little slower to get sequence information, but it now is available. And so we're developing a molecular barcode for Plasmodium vivax. In fact, Plasmodium vivax is slightly more diverse, maybe 2- or 3-fold more diverse than Plasmodium falciparum. And so it actually is easier to find polymorphic sites that have high minor allele frequency. And so these are just the chromosomes of the Plasmodium vivax parasite. The gray are all the possible alleles that meet our criteria for making a barcode. And the pink ones are the ones we've chosen based on sort of some technical details. And here are the first samples. This is hot off the press. So these are samples. These are known strains where we have the sequence. And then these are from Brazil, actually. These are the first ones we did. These are the chromosomal positions, and you can see very nicely we're getting allelic discrimination between strains, and we can get a unique barcode. You can see in certain allelic positions, we perhaps won't have so much discrimination in certain populations. So for example, this particular single nucleotide polymorphism is identical in all the strains from Brazil, as is this one. So again, we're in the process of developing this, but we now know that we have allelic discrimination and the assay is working. So we are able now to begin to ask questions about Plasmodium vivax in a very similar way using the similar tool. Okay, and I think with that, I'm going to end. I do have another, sort of whole section, but I figured I would run out of time. So one of the other things we're doing, and I'll just introduce that and then take questions, is really, this is much more practical in some sense. You all know this, that the current situation, although ACTs are working, we know there's resistance in Southeast Asia or delayed clearance. I just showed you data which tells me there will be resistance in Africa. So I think the challenge is really making new drugs and being clever about it. And I just want to say that one of the things that we found extremely valuable and something that I think I wouldn't have guessed would work is that in fact we now can begin to understand the target of any small molecule in the parasite simply by using genetics. It's relatively straightforward to select resistant mutants, and because we can start with a cloned parasite, we can just sequence the mutant genome and find the target. And we've now done this for a very large number of small molecules. It's very simple. You just grow the parasite in culture with the drug, select resistant mutants, clone them, and sequence them. And this is just a partial list, and we get different targets. These are compound classes. We get resistance, and we get target ID in almost all of these cases. So this is an incredibly powerful tool, and one that I think gives us now an opportunity. And this isn't limited to drugs. We could also select biological variants in exactly the same way, things that invade a different antigen presentation or different receptor presentation on a red cell. So I think, again, the combination of the haploid genome and the ability at really low expense now to analyze genomic information is going to give us another leapfrog also in the basic biology area. And with that, I'll stop and take questions. Thank you.
Thank you very much, Dyann. So now we have about 10 to 15 minutes for questions, not only from the malaria community, by the way, but from all of you here. So, thank you.
Dyann, that was very interesting. Thank you. There's some information from the genetic biology of arboviruses that there is less variation than would be expected for other RNA viruses. And the idea is that the alternating life cycle between a vertebrate and an invertebrate host actually creates a restriction on the variation of those organisms. And the information so far seems to suggest that there is purifying selection in vertebrates and diversification in the mosquito for those viruses. So this seems to be a little bit from the opposite, maybe, of what you're seeing with falciparum.
Right. And I think there are two things. One is that if you think about viral infection, the purifying selection means, it's basically an escape. It may be a single escape. Whereas in this case, the parasite is actually sort of, we know by this sort of different antigen presentation that probably each parasite in your body is slightly different from an antigenic perspective. And that allows it to persist, would be what I would argue. And in the mosquito, remember, it's basically a full reset. So now what's happened is you now get recombination. Remember, arboviruses, right, don't have sex, right? So the sexual exchange actually kind of resets everything, and so you never get out in a natural circumstance where you have outcrossing. You don't get back the same organism. Now, as we move to a situation where the majority of samples are actually a single parasite, if a single parasite begins to predominate, I predict that that will be the equivalent to inbreeding in other eukaryotes or even in prokaryotes, and you will see a natural loss of fitness in that population. But I think the reason it's reversed is because the sexual exchange at each transmission gives you, basically you reassort, so you don't get back the same thing, and that creates diversity. And if you think about it, every cross creates 4 progeny, which are different from one another, so that in a mosquito that has 3 genomes in it, you get 16 out. And you multiply that over a population.
Okay, more questions?
Well, so thank you very much. You showed this data showing that the genomic diversity may change rapidly in a few years, and then your interpretation is that it's probably malaria control activities that may introduce.
I remember, yes, I said it's coincidence.
Yeah. But then can you exclude some type of selection pressure, like short-term climatic changes or things like that? Is any?
Well, right, we don't know. I mean, by looking, one of the fascinating things about this is by looking at signals of selection, you can measure the signal of selection, but you don't know what the selective agent is. You have to kind of hypothesize, and then in some cases we can test. So in the case that I showed you of the PFMDR1 gene, where you're seeing basically almost 100% prevalence, I can just go in now and do a genetic exchange and ask the question, does that change susceptibility to artemisinin or lumefantrine, right? So I can test that hypothesis. But in the case of this bottlenecking, we don't know what it is. What I would say is it has to be something which is changing, well, we know it's changing the effective parasite population size. So we can calculate, this data implies, and if you calculate it, the effective population size of Plasmodium falciparum in this site in Senegal is less than 10. So it's a very small, it's a bottlenecking population. So I think that the mechanism is a population bottleneck, but what drives that bottleneck, I agree, could be dramatic, climatic change. It could be changes in mosquito species. If you're selectively controlling Gambi, Phenestis also transmits in this area. Maybe the answer is, and we haven't looked, that actually what's happening is we now have Phenestis being the major species that transmits in this area, and they're only biting a small number of individuals. Okay, so there are lots of possible explanations, but it now gives you, something is happening. Now you can go out and look.
So probably you can learn a lot if you are able to have concurrent data from different geographical areas.
Exactly.
To show these changes
Yeah.
in areas with intervention, without
Exactly.
looking to climate change, things like that.
Right. So, for example, I didn't have time to show the data, but if we do a similar experiment in Malawi, where transmission is extremely high, we never see this phenomenon. All the parasites are different, and people have multiple parasites. So I think that's exactly right, and we'd like to do it on a larger scale.
Well, I'm not in the field of malaria,
Yeah.
or immunology, nor in genetics.
Okay.
So it's a very simple question. I found very interesting that one of the bottlenecks was when the Plasmodium lives in the mosquito. And really, it seems that there is much more possibility to have success if you act at this time. So is there research ongoing, like the one you showed? You basically showed how the mosquito behaves in the individuals and how the exchange of genetic information. So do you have this type of information within the mosquito, trying to stop the infection, not when it's in the man, but when it's in the mosquito?
Right. So there's a significant amount of effort. The mosquito's interaction with the parasite is not benign, right? So there are innate immune responses in mosquitoes that limit the ability of the parasite to replicate. It's kind of the oocyst becomes melanized, and then can't develop. So in fact, there's a reasonable amount of effort in the field to try to breed refractory mosquitoes, and then, you know, those are potential, you could potentially release those. We just came from a lecture on the idea of creating sterile males so that you'd actually reduce, just like you do for medfly or screwworm, you reduce the population of mosquitoes by release of sterile males. Okay, so there are lots of strategies around that. It's very hard to do this kind of genetic analysis from the mosquito, although we are now in a position to try that. The challenge is that there are relatively few parasite genomes relative to mosquito genome in a mosquito, and it's hard to separate. Yeah. In the case of humans, because we can separate away the white cells, we actually, red cells don't have any, in humans, red cells don't have any nucleic acid. So it's relatively straightforward to get almost pure parasite DNA directly from a patient. That's not true for a mosquito, but there are technical ways around that. So yeah, so we'd love to do that. And you know, now, almost 20 years ago, Lisa Ranford Cartwright actually painstakingly dissected out oocysts, you know, so basically the tetrad product, and measure. That's how we know there are 4. So you can do it, but it's incredibly labor intensive.
I found it interesting that you propose to use the level of inbreeding as an estimate to measure transmission, but if you do that, aren't you looking many things at the same time, because you are assessing current transmission, but also previous bottlenecks, previous population size, selective sweeps, as you mentioned in another part of your talk. So how would you envisage measuring transmission just from the inbreeding level?
Well, because if, you know, you will get these clonal populations appearing in more than one patient, right? That tells you that you're really down to a low level of transmission, right? So once you have outcrossing, then all of those other things participate. But in the case where you have essentially, if you think of the N case, like in Panama, you have a single predominant genome, then all you're measuring is you're essentially measuring transmission. Transmission is down to basically as low as it can go. Yep.
In this study in Senegal, those parasites, do you have any phenotypical result of those parasites? Are there more or less virulence?
Right, so it's very interesting. Now the question is, are these parasites being selected for some biological property? They must be, but we don't know what it is. So they're not, well, you know, virulence we can't really measure because we can't test it in patients. But if you look at growing in vitro, they're fine. A fascinating result, which is not on slides yet because it hasn't been repeated. It turns out that all of the members of a group seem to express VAR genes from the same class, right? Different VAR genes, but VAR genes from the same class. So is it that VAR gene class actually is driving it in some way? Too early to tell. The data is still, but I mean that's a possibility. You know, there has been a proposal, for example, that certain classes of VAR genes lead to biological properties related to severity of disease, and you know, maybe that's what we're seeing. But I would say it's too early to tell. We don't know. We're measuring gametocytes. I think one possibility is that they actually make more gametocytes, right? They're more efficient making gametocytes. And so that's another thing that we're testing in vitro, but I don't have any data on that. So there's lots of ideas, and you know, I have to say, I have to admit a certain, you know, sort of myopic view of the world. So, you know, it never occurred to me to think about like epigenetic regulation in these identical parasites, because of course it's the perfect experiment. Here you have the same genome, so you're completely controlled at the genomic level. So now you can ask what epigenetic drivers are there in different patients. And presumably, because we select, you know, we have discovered these parasites in patients at the beginning of the rainy season in September all the way through December. What that means is it's not just a single transmission, it's probably multiple transmissions. So this is actually, we actually have parasites. We don't know their exact history, but we know they differ from one another in the number of times they've been transmitted. And so this is probably a goldmine for understanding epigenetic regulation derived from the natural setting. And as you saw in that expression data, clearly there are things that happen in patients that we don't see in vitro.
Okay, we have time for a couple of more questions. Jody?
I cannot refrain from making another question. It's, well, seeing how much you know about the genomic of this parasite, one would expect that you are very close to identify whether there are genetic determinants in the parasite that are closely related to severity of the disease.
Yes, I think that's right. I mean, I think actually, if there are parasite genomic determinants, and I will tell you one study actually that we've just completed, has been published in Malawi, where we actually compared severe patients with patients who had malaria and died of other causes, right? So this was an autopsy study. And what we discovered, this is with Terry Taylor and Dyann actually did this work, is that in patients that have uncomplicated malaria or have or die of some other cause, like an automobile accident, they have a really high diversity of parasites, not only in the bloodstream, but in all of their organs. In those patients that died of severe disease, primarily cerebral malaria, they have a predominant clone, a single clone, in many cases. And what we think that may indicate is that, in fact, what's happening is there's a clone that's taken over and overrides this partial immunity. And so it may, but those clones are not the same. So in each patient, it's a different clone. So we think maybe it's not that there's a severe disease genotype, but in fact, it's related to the patient's exposure history. And when they're exposed to a parasite type where they haven't seen it before, so it's like a new serotype, if you think about it in a serotype sense, that you can get this fulminant infection. And that's what happens. There's a precedent for that in Malarian pregnancy. So in primary pregnancies, the first pregnancy of women living in endemic areas who have this partial immunity, so they no longer get sick, they're not at risk of dying, all of a sudden there's this fulminant infection in pregnant women. That's why they're at such risk. And that turns out to be the selection by the placenta of a parasite expressing one of these, a single version of this surface antigen, which is there at very low prevalence in the rest of the population. This is the VAR2CSA gene. And in fact, the hypothesis it's a fulminant infection because the immune system in that first pregnancy doesn't recognize VAR2CSA. By the second and third pregnancy, the problem is much less. And by the fourth pregnancy, the problem goes away. So I think actually it may be that severe disease is actually related to the patient's historic exposure to different serotypes. And if you think about influenza, it's just like a pandemic influenza, it's just as an individual. I mean, that's all speculation. Everything I said from the time I started answering your question is complete speculation. Well, the data is correct, but the rest of the interpretation is speculation. But I think it's something to think about. So there probably isn't a genotype to severity. It has to do with the patient's previous exposure.
Sorry, so if that's true, so if there's lots of clonal infections, then there wouldn't be any recombination within the mosquito, right?
Exactly.
So, but doesn't that also mean that you can't necessarily infer transmission intensity from the amount of whether one patient has the same clone as another one? Because in that case, you might have a very high transmission intensity.
You could, yes, you're right. You could have a high transmission intensity, but in general, in places where there's high transmission intensity, you have multiple clones. And I think, so I mean, you could imagine a situation, yet to be identified, where you have very high intensity of transmission, where it's a single clone. I think biologically that's very unlikely. I think that these things are going to burn themselves out by inbreeding very quickly. If that happened, I would guess that parasite would go extinct. And we see some evidence for that. If you look, as we look over time, the parasites that get to this large prevalence in the population start to disappear, and we have evidence that there's increased antibody recognition of those parasites in patients. So I think what happens is when you get in like a mini epidemic, then you're going to get antibodies. I think it's like flu. I like to think about this like flu, so that you probably will never get high transmission of fungal parasites, would be my guess, but we'll see. You could be right. Theoretically, that is correct, and that's also, you know, sort of, I guess, an answer to your question. I mean, it is possible that this isn't directly a measure of transmission intensity. It's a measure of the clonality of the population.
Last one.
Last one. Then in many African countries where malaria is endemic, we have also a lot of HIV, especially in East Africa. Do you think that this can affect the evolution of the parasite, the development of the systems?
It might. I mean, you know, there's a reasonable now investigation of that, and I would say the effect is not dramatic on either infection, which, you know, I think is interesting. You have higher parasitemias and you have higher viremias in co-infected patients, but it's not a dramatic effect, and I think it probably means that CD4 immunity isn't so important to the controlling of the parasite until you get down to a very low level. And my guess is that the patients that have very low CD4 count in the days before they were being treated just didn't, malaria, they died of something else before malaria became a significant problem. But it's, you know, we haven't been involved in those studies, but there have been a number of studies published where there's just not a dramatic effect. There is some effect, but it's not overwhelming. So I don't know if it will drive it. I think probably not. I doubt if it's not bottlenecking the parasites, it may be allowing a little extra expansion.
Okay, well, thank you very much, Dyann. It's been a great lecture, and I think it helps both focused thinking, but also hopefully lateral thinking for those of you who are not so much involved in malaria. Thank you very much. I think you did very well in our purpose. Now, we will continue with the Axis Global infectious cycle outside here, where there will be some snacks. And thank you all for coming.